Dr. Perlman. Our next speaker is Dr. Dave Jacobi. Dr. Jacobi has a long personal and professional history working in the area of Friedreich's ataxia. He's a neurologist and is currently the Executive Group Medical Director of Marin Pharmaceuticals. He's the Global Clinical Development Lead for several early and late-stage programs, including the recent global registration of a drug called Brineura for the treatment of a specific form of Batten's disease, which is a rare inherited disorder of the nervous system, and the development of histone deacetylase inhibitors, or commonly known as HDACs, for Friedreich's ataxia. Dr. Jacobi has had leadership roles in another disorder called PKU, the Batten disease program, gene therapy, and the neurogenetics therapeutic area. He has 15 years experience in the pharmaceutical industry, and he's led clinical and regulatory efforts for several development programs, from translational research all the way through to new drug application filing with the FDA. Before joining Marin, Dr. Jacobi spent 8 years at Repligen Corporation. He headed programs in spinal muscular atrophy, which is a motor neuron disease, and that led to the successive collaboration agreement with Pfizer. He was also involved with a small molecule disease program in Friedreich's ataxia and biologics programs in pancreatitis and bipolar disease. David comes from a background in academic neurology from Mass General Hospital and Harvard University, having done his residency in adult neurology and a clinical fellowship training in movement disorders and basic science, fellowship in molecular neurogenetics. He received his MD and his PhD from the University of Pennsylvania and has been a pre-doctoral fellow at the Scripps Institute and received his undergraduate degree at Swarthmore College. Please welcome Dr. Jacobi.
Sorry, I was a little grandiose in my own bio. It just means I've been doing this for a long time, basically. That's the gray hairs and the nothing on the top of my head. What I'd like to talk about today is just a little introduction to Marin and what we're doing in terms of new drug development opportunities in the space of Friedreich's ataxia. And try and convince you that we both have a great program in Friedreich's ataxia. And as a company, we're leveraged to really make quick progress in terms of the development milestones for the disease. I suffer a little bit from geekdom or nerdity. If what I'm saying doesn't make any sense, just please raise your hand and I can take a step back and try and make it a little less confusing. It makes sense to me, but sometimes the rest of the world look at me like I've got two heads. A little bit about the company. So Marin specializes in single gene disorders, particularly in rare diseases. And so we have 6 marketed drugs and 5 diseases in the pipeline. But the consequence of that is we are truly kind of a fully integrated biopharmaceutical company that treats patients around the world. So we have infrastructure and support in terms of finding patients, supporting patients, doing the clinical trials, and distributing our medicines in all parts of the world. Many genetic diseases, as Dr. Perlman just explained, actually have different incidences or prevalences in different parts of the world, and so we are truly a global, worldwide development organization. We've been growing. We're about 3,000 people, and that's about half of that is research, and about half of that is supporting the marketing of our approved drugs. It's a complicated slide, but basically the sort of the funny purple things on top are the approved medicines. And the diseases that we tend to work in are very rare, but like Friedreich's ataxia, they're single gene disorders for which the mutation or what causes the disease allows a biology that can be easily treated. So many of our drugs are lysosomal storage disorders. There's a single enzyme that lives in a part of the cell called the lysosome. And if you can replace that enzyme, you can significantly alter the trajectory of the disease. So the NPS disorders are all lysosomal storage disorders. We have a small molecule Kuvan for PKU. PKU, again, is an enzyme that breaks down phenylalanine, which is an amino acid. So when you look at a Diet Coke can, it says, beware, it contains phenylalanine. It's for those people that don't have the enzyme that can break it down. And so we have a molecule both approved and a molecule in development for the treatment of PKU. We had a recent approval for Batten's disease. This is an interesting disease because this is a pediatric neurodegenerative disease, and it's a very severe disease. But this, again, is also a single gene disorder. It's an enzyme deficiency, and we were able to determine if you actually give the enzyme directly back to the brain, the disease is significantly altered. And so these are small children now that have stabilized neurology and have a very good quality of life with that enzyme replacement therapy. Our pipeline is built out basically, again, with the same sort of theme. And so you'll see we actually have a new molecule that's in review now at the FDA, and we're just about to submit the licensing agreement in Europe for PKU. So we're trying to get a broader treatment population in PKU. We just dosed our first phase 3 patient for the gene therapy trials for hemophilia. So we're interested in new technologies. We don't just make audio visuals, not my strong. We don't just make protein biologics, but we have small molecules, we have oligosense molecules, and we have a gene therapy platform that's allowed us to learn a lot about how to engineer a vector and how to manufacture a vector. Great. Thank you very much. And so we have another experimental program in a lysosomal storage disease called MPS IIIB, which again is a pediatric neurodegenerative disorder. And then the last one on the slide there, BMN 290, this is our HDAC program for Friedreich's ataxia, which will be in the clinic at the end of the year.
Yeah.
So, not full time, I realize. So we wanted to highlight the fact that we're interested not so much in the type of technology, but the type of disease. And we're willing to leverage different technologies in order to solve the problem. And so hemophilia is a clotting disorder. It's also a single gene disorder. The mutation means that you don't make one of the factors that you need to have in order to clot your blood. The nice thing about hemophilia as a potential therapy for gene therapy is that small changes in the amount of factor VIII, which is the clotting factor, lead to big changes in the clinical outcomes. And that you can target the liver and you can make factor VIII that gets into the bloodstream. So challenges for different diseases are basically, can you get it to the tissue that is diseased, and can you make enough to make a change? And we're learning a lot about the gene therapy space by going into hemophilia. And the other thing I just wanted to say, that dovetails very nicely with Dr. Perlman's talk, is that natural history is an incredibly important part of what we do in rare diseases. This graph just shows that the average time it takes for BioMarin to go from filing an IND, which is the permission we get from a regulatory agency to do a clinical trial, to actually approval of that drug for treatment as a commercial entity is about 4 to 5 years. The space between first patient in and approval for Brineura for CLN2 disease was 44 months. We have a lot of leverage because these are diseases for which there is a high unmet clinical need and usually not a lot of treatment. But there's also these issues about placebo and utility to the patient that the regulators understand, especially in these disease spaces. And so we can leverage natural history to really design very efficient clinical development paradigms. Usually in diseases that we were involved in, BioMarin needs to come in, and while it's a research program, we need to invest in natural history so that we learn what the untreated course of disease is. Friedreich's ataxia, this room, you guys have been doing this now for nearly 20 years. And so the amount of data that's available to a drug developer to exploit in terms of understanding both endpoints and what the course of untreated disease is very, very large. So I can't emphasize enough what Dr. Perlman said, that your participation really it allows us to be incredibly efficient in drug development. And so it makes a really big difference in terms of the length of time it takes us to approve a drug. I think every company that probably talks to you says these sorts of things, but we do really get a lot of excitement after of just kind of understanding you as a patient population. And always in rare diseases, the space between patients and the company is very small. And so this slide is kind of a corporate propaganda, but I would always say, please never hesitate to reach out to BioMarin and to me personally, because we really feel like if we can't understand what the needs of the patient community are, we can't develop a clinical development plan that really is going to leverage what the unmet needs of the patient population are. So let me pivot a little bit. That's the company, and that's why we're really interested in Friedreich's ataxia as a disease to do drug development in, and talk a little bit about our first foray into a therapeutic paradigm for the treatment of this disease. This is a slide that I stole from Dr. Massimo Pandolfo in Brussels. And the point is a lot of words, but the point that I would like to make here is the same point that Dr. Perlman made, is that the disease is simple in that it is a disease of deficiency of frataxin expression. We don't make enough frataxin basically to support mitochondrial function in certain disease tissues. And so if we can turn frataxin back on, we can substantially modify the course of the disease. It's basically the paradigm a number of different drug development companies are going after, but in different ways. The thing that's really valuable is not only is there a mouse in which there's a dramatic change in both turning on and turning off the gene, but there's also natural history. So we know that if you're born with, say, an average of 600 repeats in the first intron of your frataxin gene, you will have typical onset of Friedreich's ataxia. You will express about 15 to 20% of the normal amount of frataxin in every cell of your body. And that's the disease that classically comes on in adolescence and results in ataxia and potentially further, more serious problems down the road. But there are people that have less repeats who have later on stage disease, and they tend to express a little bit more. Not that much more, usually about 30 or 35% of the normal level. And then there are carriers, which are, we call them truly haploinsufficient, but it means if you have one copy of the mutation, you're going to be absolutely normal your whole life, but you are going to make about half as much frataxin. So the point here is that if we could double or triple the amount of frataxin in our bodies, we think we can have a meaningful clinical response and modify the course of the disease. And that's basically the basis for why we think HDACs might be a valuable tool for modulating the expression of frataxin. So the reason we think that is represented here in this cartoon. So there's basically two examples here. One is a gene that doesn't have an expansion in the first intron, and the bottom is a gene that does have an expansion in the first intron. So there's a couple of really just simple points that I'd like to make that are really important. First is the mutation that occurs in the gene doesn't affect the sequence of the gene. And actually, all it does is it turns it off. And so this is really important because the gene itself doesn't need to be changed. The gene just needs to be turned on. And so what the mutation does is it sits in actually a part of the gene that doesn't get turned into protein. It's actually sort of we call it an intervening sequence. It exists in the DNA, but it doesn't exist in the messenger RNA or the protein. But what that mutation does is it turns off gene expression. So it actually recruits in proteins that make the chromatin very compact, and you initiate expression, but you can't read through the gene. So what you see on the bottom here is the fact that the expanded GA repeat allows initiation of expression, but once it hits that big repeat, it stalls. It falls off the DNA. It can no longer be expressed. And so you have a very inefficient gene expression that knocks down normal gene expression to about 15 to 20% of the normal level. The hallmark of that kind of altered chromatin is that it's very dense It's very compact, and it doesn't have some of the markers that allow it to open up and be expressed. And so one of the things that I'd like to explain a little bit is the molecules that we're working on actually change the topology of the chromatin, open it up. It allows the gene expression to read all the way through the frataxin. So all we're doing is we're taking the abnormal DNA that's the result of the mutation and opening it up and allowing the transcription process to go forward. We call this epigenetics because we're not changing the DNA, we're just changing the structure of the chromatin so that you can have read-through on the frataxin gene. And the cartoon on the top left kind of gives you an example. DNA in your chromosomes are actually wrapped around protein, and if they condense, that's sort of what we do to store it so it can't be expressed. But if you have a gene there and you want to make the gene, you have to open it up. And so the mutation in the frataxin gene causes it to be condensed like it is on the bottom part of the top left. And the drug that we're trying to develop opens it up into the state that you see on the top. These are complicated diagrams, but this basically shows in the top right that the amount of frataxin that is made in diseased tissues like the cerebellum is substantially reduced compared to a carrier or a normal person. And the graph on the left shows that acetyl status of histones, so histones are the packaging proteins, and acetyls don't like each other very much, so if you take the acetyl groups off, the histones compact. If you put acetyl groups back on, the histones open up. And so the acetyl status of the mutated frataxin allele is very small. There's no acetyl groups there, and the DNA is very compact. And so a histone deacetylase inhibitor is to try and re-put acetyl groups onto those histone molecules and open the histones up. And lastly, the graph on the right says that the protein expression of frataxin is something that we can leverage as a biomarker. So the red lines on the bottom are patients that were serially phlebotomized every week for 13 weeks. And we measured their frataxin in peripheral blood lymphocytes, which are cells that circulate in the blood. And we compared it to a related sibling that didn't have the disease. And you can see that there's always that separation in the amount of frataxin. And the goal of treatment is to take the level of frataxin that exists in the red lines and bump them up to the level of expression that exists in the blue lines. Because we know the blue lines have no disease for the entire course of their life. So how do we do this? DNA is in a dynamic state. If you need it, it's open. If you don't need it, it's compact. And there's basically two sets of enzymes that regulate the way the DNA is in. One is called an acetyltransferase, which puts acetyl groups onto the histones, and one is called a histone deacetylase, which takes them off. If you inhibit the deacetylase, which in this case is the eraser, you can leave chromatin in the open state. So a small molecule that allows you to inhibit that specific deacetylase that compacts frataxin will allow frataxin to be in an open state and to be read through. So this kind of summarizes the stuff that I've been saying, but we have a small molecule that is something that does inhibit histone deacetylase that we think is a valid treatment entity for Friedreich's ataxia. We think that because the mutation causes abnormally condensed chromatin, and we know the molecule can open it up. There are questions associated with these treatments, however. So company guys like myself are pretty good at telling you about how they're going to work and the optimistic parts, but HDACs work in the entire genome. And so they can cause gene dysregulation in other places besides the frataxin locus. So the questions that we have and that you should have about HDACs are, firstly, there are, as Dr. Torrance says, different sites in your body where the mutation is causing problems. So the molecule needs to get there in order to change your DNA. So what if the molecule doesn't get into the heart? What if it doesn't get into the brain? Even if it gets into the brain, what if it doesn't get into the cerebellum or the spinal cord or the dorsal root ganglia? So there's always issues about tissue penetration The other part is whether there are bystander effects. So if it's a histone deacetylase inhibitor, what if it causes other genes to be dysregulated? Is that going to be a bad thing? And is it really a histone deacetylase inhibitor, or does it do other things? So parts of those questions have been answered already, but parts of those questions are really what we do when we're doing clinical development. So there are molecules like, for example, valproic acid, which have been approved for a long period of time, that have lots of gene expression changes and have been shown to be safe in a number of indications. HDAC inhibitors themselves have been approved mostly for the treatment of cancers. And so the real challenge here is to find a molecule that's much more specific for the frataxin locus than it is for other genes in your DNA. So we have a molecule that we're calling BMN 290, and I've already gotten some criticism from Kyle about the fact that it's not a particularly sexy name. But this is our clinical candidate. And if I was just to take a step back, I think many people in the room know that these molecules were actually discovered at Scripps Clinic by Joel Gosfeld. And he actually did an experiment where he put a number of different molecules onto cells from patients that had Friedreich's ataxia. And he noticed that one specific class of molecules seemed to have specificity for the frataxin locus. In other words, they didn't cause widespread gene changes, but they acted not on the normal locus, but on the mutant frataxin locus. And that discovery is what actually was in-licensed into Repligen. And Repligen modified the discovery to something that had slightly better pharmacologic properties that allowed them to do a study, a phase 1 study in patients in Italy. And this is published by Liz Saragni in the Annals of Neurology, I think in 2015. We learned some things there, but it turned out to be a false start. We learned that you can take the molecule by mouth, and a single dose of the HDAC inhibitor would increase the transcription of frataxin in peripheral blood lymphocytes, which is a great principle. But what we also discovered is that molecule didn't penetrate the tissues particularly well. And it had a problem with its breakdown, so that there was a metabolite that was made that was potentially harmful. So it was back to the drawing board. And so when I moved to BioMarin in-licensed that program from Repligen, and it's been in the lab basically undergoing medicinal chemistry. And the molecule 290 now is a molecule that does get into the tissues, both heart and brain, and I'll show you some of that data. And also has solved, we think, the metabolic liability of the original molecule. So we're happy that we did the clinical trial because it showed the principle of treatment with HDAC inhibitors is a valid approach to treatment. And now we think we have a molecule that can escape some of the side effects that we saw in the first study. Okay, this is basically making the same point, but one of the things that I'd like to just basically drive home here is the small molecule that we pulled out, 290, gets into the heart and to the brain and all parts of the neuroaxis about 2 to 3 to 4 times better than it is in the periphery. So the molecule actually seems to sort of home to the tissues that are affected in the disease. The other thing I'd like to say is that it's not the same kind of HDAC inhibitor that exists in the clinic for oncology. And that it affects, there's 12 different forms of HDACs, and it's quite specific for just one form. And that one form seems to be responsible for controlling the frataxin locus. So this is just an experiment that shows you couldn't use just any HDAC inhibitor on the frataxin locus. So the bottom panel is a drug called Varinistat, which is actually approved by Merck for the treatment of certain oncology indications. It's very good at reacetylating histones. So you see the dark band in the middle panel on the bottom, that's reacetylating histones. But you see in the bottom panel that frataxin doesn't change very much. If you look at a related molecule to 290 on the top panel, you see that it reacetylates histones, so it should change expression, but the frataxin expression is upregulated. So there are specific HDAC isoforms that seem to control the frataxin locus. And if you can inhibit those, you can upregulate frataxin without causing disturbances in other genes. And this slide also just basically demonstrates that the drug has a high predilection for the brain and the heart in not animal studies, but in not in humans, but in animal studies. But the nice thing for us is it's predictable in multiple animals, and so we believe that it's also going to be true in humans. So if you look on the left panel, you see the doses by mouth into a rat basically result in better PK compared to the original molecule. The original molecule is in blue, 290 is green, so it lasts longer in the bloodstream. And if you see that the dashed lines are actually the concentrations in the brain. If you look at, in the middle panel, the distribution in other tissues in the rat after oral dose, you see that plasma is the purple line on the bottom, but the drug is even more concentrated in the heart and in the brain. And then the panel on the right is my point that distribution is stable across species. So our pharmacologists tell us that it's very likely to be the same in humans as it is in animal studies. And this is the point I was trying to make before, is that the molecule that we've chosen for the treatment of FA isn't something that inhibits all HDACs, it actually inhibits a specific kind. So if you look at the TSA molecule, that is something that inhibits all HDACs, and you see the smaller numbers are more potent. And it's very potent on 1, 2, 3, and 6. 290 is most potent on 3. And it's actually not a particularly potent HDAC inhibitor, which again, we think is great because it's potent at the frataxin locus, but not across the entire genome. And it's specific for 3 and not for 1, 2, and 6. And then lastly, if you look at its effect on frataxin, it basically can make a twofold change in the level of frataxin in different cellular models of disease. So we have a little bit of difficulty in certain types of animal models. So the way that we've been doing screening is in human cells that have been differentiated in a laboratory petri dish into a neuron. And then we put the drug onto those cells and we ask whether frataxin can be changed. And we see about a twofold change in frataxin at concentrations that we can easily get when we dose the molecule by mouth. And it's a nice dose-response curve, so it allows us to kind of model a little bit about how much drug you need to get in order to raise frataxin a certain amount. So as Dr. Perlman said, we do know that there is some plasticity. So what I mean by that is, when you lose function, there's a component of it that's probably because of cell death, but there's another component of it that's probably because of cell dysfunction. And so we do believe that by restoring frataxin expression into the neighborhood that carriers have, we won't be able to impact the proportion of cells that are dead, but the cells that aren't working very well, when they get frataxin back, they should actually now start to improve. And this, I think, is maybe my last slide, but right now, the molecule is identified as our clinical candidate. We're done with doing research in our medicinal chemistry lab. The molecule has been tested in multiple species in terms of sort of a subacute toxicology and has passed muster. So it's now going into the final test in animal studies before we apply to the regulatory agencies for approval to do clinical trials in human beings. So the timeline for us is that those studies will be done and the reports will be ready to submit to the agency in August or September. So we expect to be able to go into the clinic in October or November of this year. The clinical development plan is going to be, I think, quick, but the first part of the clinical development plan is to determine whether the molecule is safe. And so it's a phase 1 program. And so we will do 3 clinical studies in 2019 that determine the safety of the molecule for patient exposure. And then we will do, as soon as we determine that the molecule can be dosed safely, we'll do long-term efficacy studies the year after. So that was my lesson. Thanks very much. I would be here all day. It's like jokes in the Catskills. But I realize that there's a lot of information and there are perhaps a lot of questions about it. So please don't hesitate to come up to me at any point and ask a question. I'd be more than happy to talk about the program. Thanks very much.
How was the grade called?